In this video, we're going to look at genetic engineering, otherwise known as in vivo cloning. So you've done a little bit of genetic engineering in GCSE, so it's just a continuation from what you know from there. So what is genetic engineering? It's the process of cutting out a gene from one organism and inserting it into another to create what we call a transgenic organism. So you can see why we call it transgenic. Trans means across gene. So you've moved a gene across from one organism to another. And it involves the following 4 stages that we're going to look at. So first of all, you're going to isolate the gene that you want. So it could be a toxin gene, it could be a gene for a particular protein like insulin. You're then going to insert it into what we call a vector. A vector is something that carries the gene from one place to another. So we're going to look specifically at plasmids, which are found in bacteria, but viruses can act as vectors as well. We're going to look at the insertion of the vector into the cell, in this case, the bacterial cell. And finally, how we identify and grow our transformed cells, that's cells that have taken up our vector containing the gene. So first stage is isolation of the gene. So there are 3 possible methods you need to know for isolating the gene. So first of all, we can get it from the genome itself. So that's from the whole of the genome. That's all the DNA in a cell. You could make it from messenger RNA. We'll look at the advantages of that in a minute. Or you could make the gene using the gene machine, which is the most common method nowadays. It sort of goes through 1 to 3. So 1 was the first method, 2 became the second, and then number 3 is the method they'll use now. So if we look at how we isolate it from the genome, basically what you need to do is you need to cut the gene out of your genome. So imagine a long piece of string where you only want a small part of it, a bit colored yellow. You're going to cut just that yellow bit out of the whole piece using what we call restriction enzymes. Now, restriction enzymes cut at specific base sequences. There are lots of different restriction enzymes that all cut at different sequences, and they give what we call sticky ends. So you've got an example here of some DNA, just a random set of bases that I happen to choose, a random sequence. And you can see that this restriction enzyme cuts in this sort of zigzag fashion. So you end up with 2 ends, one which is TTCTT and the other AAGAA. And you can see that these are obviously complementary to each other, which makes sense because they've just been cut that way. Now that means that if we cut 2 different pieces of DNA with the same restriction enzyme and we bring them together, they will naturally complementary base pair. So they will naturally sort of stick together. So restriction enzymes are like our genetic scissors. They're used to cut the pieces of DNA. And this is a method you can use. The problem with this is if you imagine cutting your piece of string with scissors in random places, you're going to end up with lots and lots of pieces of your string, of which you're going to have to then find which one contains the gene. So it's the same for the human genome. If you cut the whole of the human genome up using a restriction enzyme and you only want the insulin gene, you're going to have to find which 2 pieces of the whole of the genome contain your insulin gene, one from each chromosome, paternal and maternal. And there are hundreds and thousands of genes, and you only want 2, and you've got to then find them. So that's a little bit of a challenge, which is why it's a method that we don't tend to use nowadays. So the second method is to isolate the gene using messenger RNA. And this has its distinct advantages. So if, for example, I want to isolate the insulin gene, I know that in the beta cells of the islets of Langerhans, they'll be making lots of insulin. So therefore, there'll be lots of messenger RNA for insulin. So therefore, instead of just getting 2 copies, I'm going to get thousands and thousands of copies. The other thing that's worth knowing is that messenger RNA does not contain the introns because they've been spliced out already, which is really, really useful. So the bacteria don't need to sort of know what to do with those introns. So the first stage is that we use the enzyme called reverse transcriptase. Now, you should have come across reverse transcriptase. It's found in HIV. Its job in HIV is to convert the RNA found in HIV into DNA so that it can be incorporated into the genome. So we are basically taking that and we're using that information. So we are using reverse transcriptase to convert messenger RNA into a single-stranded, or to make a copy of the messenger RNA as a form of single-stranded DNA over here. We will then basically destroy our messenger RNA strand. So we've now just got a single piece of DNA, which we can then copy using normal DNA polymerase to get what we call cDNA, shown down here. And finally, we will add to that cDNA our promoter sequence, our terminator sequence, and our sticky ends. So we've come across the promoter sequence. It's where transcription factors bind to allow transcription to be activated. And obviously you need terminator sequence that tells it where to finish. And you've got sticky ends because we're going to need to put them into our vector. So you're going to have to add them all on once you've made your cDNA. But as we said, this is a good method because you've got lots of messenger RNA and you've got no intron. So it's a very convenient method to use. The third method is really, really simple. It's to use a gene machine. So you basically send the full base sequence, including the promoter, the terminator, and the sticky ends to the manufacturer. The manufacturer will use a gene machine and produce a gene and send it back to you. And that's obviously the more modern method. If, however, you're asked to describe in an exam question a method of isolating the gene for like 4 marks, I wouldn't use a gene machine because there's very little to talk about. Whereas for 4 marks, it's nice and easy to describe this process. You can see very clearly how you're going to get the marks. You're going to get marks for reverse transcriptase. You're going to get a mark for what reverse transcriptase does. You're going to get a mark for DNA polymerase and what it does. You get a mark for adding the promoter, the terminator, and the sticky ends. So if you are choosing in an exam question to choose a method for isolating your DNA, I would choose either method 1 or method 2. Don't choose method 3. But if you were describing it in an essay, you might mention all 3 methods, or if you were to ask to name some methods, then you could obviously mention any of those methods. So the next thing we're going to look at is how to insert your gene into your vector. In this particular case, we're going to look at using a plasmid, which are the small extra-circle pieces of DNA found in bacterial cells that provide things like antibiotic resistance. So you're going to have on this plasmid a marker gene. There are a few different types, but fluorescence and antibiotic resistance are 2 types. You're not expected to know specific types. You are expected to know their function. We'll come to that in a minute. And then you have a space on the other side where your restriction enzyme will cut. So the idea is that we're going to cut the plasmid with the same restriction enzyme that we used before to isolate our gene. We're then going to mix our cut plasmid with its sticky ends with our gene with its sticky ends, and they should go together with a complementary base pairing. That's not going to be permanent. So we're going to add DNA ligase, which is basically genetic glue. And the job of the DNA ligase is to create those phosphodiester bonds, permanently joining our plasmid to the gene. We now call this a recombinant plasmid. Basically, it's recombined. It's combined the original plasmid with our particular gene. So we then got stage 3, which is to insert the recombinant plasmid back into the bacteria. So here are our bacterial cells. Here are our recombinant plasmids. We want to persuade the bacterial cells to take them up. We need to make the cell walls permeable. We do that by treating it with calcium ions and with heat shock. Heat shock means literally heating up for a matter of seconds, then cooling it back down. And some of the bacteria hopefully will take up our plasmids. So you can see that the 2 bacteria in the middle and on the left, they've been what we call transformed. They have taken up my plasmid. The one on the right, however, has not. And the problem is we will now need to isolate which ones have taken them up and which ones have not. And this is the purpose of that marker gene, because let's imagine that marker gene was fluorescence. We now know that the 2 in the middle and the left will fluoresce and the one on the right won't. Or if it's antibiotic resistance, the middle and the left will be resistant to an antibiotic and the one on the right will not. So we can identify which bacteria have been transformed using our marker gene. That's the sole purpose of the marker gene. And then we can grow the bacteria and isolate the product. So what do we use it for? So they're not going to ask you for specific uses, but they will ask you to evaluate uses. So we know we use it for things like making proteins, insulin, growth hormone, for example. We use bacteria for that. We can produce genetically modified crops using bacteria that infect crops, making them drought resistant, weed killer resistant, pest resistant, increase their yield. We can produce genetically modified animals by inserting DNA directly into fertilized eggs that are disease resistant or have increased yield. We're looking at using milk or fruit and vegetables to produce vaccines. So getting proteins produced in the mammary glands of a cow or produced in a particular fruit like a banana, for example. Now, there are concerns over genetic engineering, and again, you need to sort of evaluate from the question, but a few things just to consider. People worry whether they're safe for human consumption or whether they have long-term effects that we don't know about. They're worried that the gene will spread to wild organisms, so you might end up with weed killer resistant weeds, for example, from cross-pollination. They're worried about the environmental effect on the food web. So if you make a plant pest resistant by producing a toxin that kills the animal that tries to eat it, then obviously you've then got a knock-on effect on the animal that eats that primary consumer and so on. So you've got effects on the whole food web because you've altered something lower down. So just some things to consider there when you're thinking about the ethics of genetic engineering. So that's a quick summary of the process. As I said, the main process you need to be able to describe is the process using bacteria, and then just evaluate it with that.